seminar remotely. So Cindy, should I get started with the introduction?
Yeah, you can go ahead.
Okay, so everybody, welcome today to the Botany Seminar Series. It's my pleasure to introduce David Domoszych from Skidmore College in Saratoga Springs, New York. So for those of you who were here, got to hear David talk a little bit about Saratoga Springs. It's a beautiful town that matches David's keenly honed aesthetic sense. I mean, his science is beautiful, and I saw it firsthand myself at a cell wall meeting in California. And so David blends imaging and cell biology and leverages the power of evolution and comparative biology and in his focus on assembly and function of the cell wall and extracellular matrix. So he has pioneered the use of the Carifacean algae and in particular the Penium system to analyze cell wall functions from the point of secretion and assembly in the extracellular space and function in the context of morphogenesis and adhesion. And Skidmore College is an undergraduate institution, and David has published numerous high-profile papers and has involved over 100 undergraduate students in his research in the last 10 years. So it's impressive the quality of his work, and that he's been so successful in including undergraduate researchers in his program. And so his work has been consistently funded by the NSF, MCB, and competitive with researchers at Research One institutions across the country. David's a highly sought after collaborator and has published papers with the giants in the cell biology field: William Willits, Josh Rose, Georgia Drakakaki, Michael Hahn. Just an amazing scientific record. And I'm just really pleased that you could join us today and share with us some of your research so people can learn about what you've done. So everybody, if you join with a virtual clap and a welcome for David, but really excited to hear about what you have to say today. So thank you, David.
Sure thing. Well, Michael, thank you for the very nice introduction, and thank you all for inviting me to present this seminar, which has been a passion of my professional life, working with these algae. And today I'm going to tell you a little bit about the streptophyte algae, or also the charophytes, as they are also commonly known. And this is the group of green algae that are most closely associated or most closely related to land plants and whose ancestors gave rise to land plants. Okay, so who belongs to the streptophyte algae? Well, if you went back about 50, 60 years ago and you were thinking about how did land plants evolve? Where did they come from? We knew they came from green algae. And at that time, people thought it might be a chlamydomonas-like organism that gave rise to a multicellular organism and ultimately to filamentous forms and a variety, and ultimately leading to land plants. But it wasn't until the 1970s and 1980s, primarily using ultrastructural evidence, that plant biologists with people like Jeremy Pickett Heaps was a pioneer at University of Colorado. My mentors, Carl Maddox and Ken Stewart, and they showed that there was a group of 6 taxa of green algae that were in a group that were most closely related to land plants. And they called them the charophytes. And I think it's more taxonomically correct today to call them the streptophyte algae. So who belongs to the streptophyte algae? Well, first of all, there are these 3 early divergent taxa, the more primitive forms. And they include things like the unicellular mesostigma, which is loaded with these beautiful scales, okay, and doesn't have a cell wall. We have the Chlorokybus group, and this is represented by the Chlorokybus genus, which forms these packets of cells, and it's found in some very unusual places. On the toe of a statue of Napoleon in Europe, also in places in the Alps, I kid you not, where people stop off and pee. So much like it's urea, as well as statues. And it's unique because it also is one of the earliest organisms known to produce tannins. Then we come to the Clepsyrmidium group. And if you've ever been to a southwestern desert and you've walked on the sand, it's not like walking on a beach. You'd be hearing a crunch, crunch, crunch sound. And that's because Clepsyrmidium forms these biofilms on desert soils, not all desert soils. And you're stepping on the dried out polysaccharide that it secretes. But from an evolutionary standpoint, it's been the late divergent charophytes or streptophytes that have really made an impression on evolutionary biologists. And in this group, we have things like Chara and Nitella, the stoneworts. We have Coleochaete, which you may have seen in a laboratory that sometimes produces parenchymatous tissue. And then we have the Zygnematophyceae, and these are the conjugating green algae. They do not produce sperm and egg. Rather, two cells come together, they form a papilla between themselves, and then you have plasmogamy and karyogamy, and that's the way they undergo sexual reproduction. Well, during the late 1990s and into the early 2000s, a lot of molecular evidence, genomic screening and sequencing the transcriptome came about looking at these organisms. And the big question was, which of these late divergent forms were the closest living ancestors to land plants? So being from a bedding town like Saratoga Springs, the even money was on the Charophyceae and on the Coleochaetophyceae because of the fact that they were multicellular. They also produce gametangia that are remarkably like Antheridia and Archegonia. And about 2010, the verdict came out after a complete genetic screening from the laboratories of Charles Dulwich at the University of Maryland. And it turned out it was the Zygnematophyceae that are the closest living ancestors of land plants. And within this group of algae, you have things like Spirogyra and its relatives, and you have desmids. And here you're seeing a picture of Microsterias, one of the most beautiful algae you're going to find. All right, so these are the organisms, the Zygnematophyceae, that were the ones that an ancestor of it gave rise to land plants. So let's go back in history. I mean, really way back in history. And we'll go about 500 to 600 million years ago. Terrestrial environments were extremely hostile. There was very little life in terrestrial ecosystems, certainly some prokaryotes, maybe some fungi. But if there was other forms of life, they would be found along the shorelines of freshwater ecosystems or along the oceans. So just to give you some comparative information, this is what some of the characteristics were, or are the characteristics of what we have today. All right, so 500 million years ago, it's believed that carbon dioxide levels were about a fourth or a fifth of, were 4 or 5 times higher today. Oxygen levels were about a tenth of what they are today. The ozone layer, because of low amounts of oxygen, was about 40% of its current level, and more ultraviolet light, deadly ultraviolet light could come through. The average surface temperature was quite warm, between 35 and 45 degrees C. And then there were other issues that were there. For example, water fluctuations in freshwater ecosystems. You had drying, you had osmotic stress, you had salt stress. And there was a lack of phosphorus that was found in primitive substrates that limited how green algae and how far they could move onto land. All right, so these were the issues that these organisms had to deal with. And there were many examples of where some photosynthetic organisms moved on to land, but it was the streptophytes, and particularly the Zygnematophytes, that were the successful ones. And it probably happened only once. So what was in the molecular toolbox that allowed ancient streptophytes streptophyte algae to come onto land? Well, looking at the genomes of several of the extant charophytes or streptophyte algae, there are lots of different things. Okay, for example, the ability to produce phenylpropanoids, things like tannins, maybe even lignin, the tannins primarily for ultraviolet screening. They developed highly developed chloroplasts with very, very distinct grana and different modes of starch production that allow them to survive in some of the harsh conditions of terrestrial ecosystems. Hormones Okay, it's been shown that hormones like ABA, ethylene, jasmonic acid, okay, were produced for surviving periods of stress. And also the interactions, they had the molecular toolbox for forming interactions with local microbiome organisms, possibly with mycorrhizal fungi. And there was one other thing that was really important, and this is where my interest lies, and that is with the extracellular matrix. What was found in the cell wall and beyond the cell wall that might have been key in the evolution of these organisms. So the extracellular matrix of these late divergent streptophytes offer considerable insight into the evolution of land plants, but also you can able to use these organisms as model for looking at some basic cell biology questions, especially those relating to the cell wall and other secretory components. So today, what I'd like to talk to you about is the dynamic extracellular matrix of our model organism, Penium margaritaceum. And then the second thing I'll tell you a little bit about is recent work that we've done with expansion, cell division, and multicellularity, and maybe dealing with and maybe early examples of cell wall integrity maintenance and phenotypic plasticity. And then I'll finish up by pointing out a couple of future directions. And knowing that there's a lot of graduate students in the audience, I'd like to encourage you to think about using these organisms as potential models. They're easy to grow, and they, of course, are not land plants, but they can provide answers to some very important questions in cell and molecular biology. So the organism that I use as my model organism, and that's Penium margaritaceum. And Penium has become a very effective tool in studying many aspects of plant cell biology and evolution. Penium is simple. It's an elongate cylinder with rounded edges. It doesn't have very unusual shapes. So when you are looking for changes, for example, in its morphotypes, it's relatively easy to see. It also will produce only a primary cell wall. We don't have to deal with secondary cell walls as we see in many land plants. And it also produces prodigious amounts of an extracellular gel. It's called EPS. EPS, the whole little biofilm polysaccharide stuff. So eukaryotes like prokaryotes can produce EPS and are also very important components of biofilms. In addition, what makes Penium valuable is the fact that you can do live cell labeling, particularly of the cell wall, with monoclonal antibodies and also other fluorescent probes. It allows you, this organism, because of its rapid growth rate, to accommodate rapid screening of various types of stress. And it has a sequenced genome. So a few years ago, in collaboration with Cornell University, the Boyce Thompson Institute, and a few other laboratories, the genome of Penium came out, and you can get it here in Cell. All right, so the plant cell wall is the most common and most well-studied component of the extracellular matrix of plants. And just as a quick review, the plant cell wall is really hydrated, fiber reinforced composite consisting of cellulose microfibrils and then cross-linked in a hydrated gel of pectins, hemicelluloses. And in the world of pectins, we have things like homogalacturonans. We also have rhamnogalacturonans. And then we have a variety of structural proteins. So what the first thing that we were going to do is, for all of the streptophytes, we wanted to get an idea as to what was the composition of the cell wall. Certainly, there was some biochemical data that was available about streptophyte algal cell walls. But about 10, 15 years ago, we worked with people at the University of Copenhagen, Bill Willits and Eben Sorensen. Now they are at different universities. And we used immunoprofiling technology, something called comprehensive polymer profiling. And this is just sort of a variation of ELISA, where you take a series of monoclonal antibodies that are recognizing specific epitopes of cell wall polymers from higher plants, and you apply them to these algae or components extracted from these algae. And you could get a kind of a picture, a snapshot of what's in the wall. So in this very, very busy heat map but I'll point out the sort of the key things. This is where we looked at three fractions of the cell wall. CDTA is something that takes out most pectins. Sodium hydroxide takes out some resistant pectins and has some hemicelluloses, and cadoxin takes out a lot of these resistant hemicelluloses. And then next to those, what you'll see, oops, sorry about that. Okay, so the next thing that you'll see next to these fractions are a list of charophyte or streptophyte algal taxa that we use. Then up on top, we have the different antibodies that were applied and what their specificity is. So when we did this general screening of these charophyte algae, what we found was, yes, we have pectins, okay, especially homogalacturonan. We have a series of hemicelluloses, including things like mixed linkage beta-1,3, beta-1,4 glucans, mannins, and a few other things. And we also found the presence of proteoglycans, specifically AGPs, arabinogalactan proteins, and a little bit of extensin. And of course, in the cell wall, you have cellulose. So looking at these results, we determined that there's really quite a bit of remarkable similarity between the cell walls of many of the late divergent streptophyte algae and those of primary cell walls of land plants. So the next step was, let's take a look at Penium. Using the same fractions, what we were able to find were things like homogalacturonan. We also found hairy pectin, rhamnogalacturonan 1 in large amounts. We were also interested in, we did some carbohydrate binding module labeling, and we found a lot of cellulose, and we'll get to that in just a little bit. And we found AGPs, extensive as well, but we didn't get into great detail in investigating them. The next thing we wanted to do, just to get a little bit better profile of what was in the extracellular matrix of Penium, was we did some work with the Complex Carbohydrate Center at the University of Georgia. And this is a heat map of the results. Now, this is where we used over 150 different antibodies with different specificities for cell wall polymers. And we looked at, and I encourage you to look at the different fractions that we have with the cell wall. We also did some work with the EPS, which we'll eventually get to. But when we look at this very extensive immuno-based survey of what kinds of polymers were found in the cell wall, once again, we find pectins. Once again, we find the rhamnogalacturonan 1 backbone. We also find RG1 and arabinogalactans, and this is a black box. And I think it's pretty much the same thing for a lot of land plants. These arabinogalactans and rhamnogalacturonan 1 and what their association is. And this is something that we are looking into now with some of our colleagues in Australia as to what we actually have here. So the important thing is that when we were doing our surveys, we have Penium that has a cell wall that is somewhat similar to what you find in the primary cell walls of land plants. When we did the genome, we then started looking for genes for cell wall biosynthesis and organization. And one of the most notable features of the Penium genome is the remarkable expansion of these cell wall related genes. And there's a lot of repetition in the Penium genome, but these are the classes of some of the carbohydrate-based enzymes that work on polysaccharides of the cell wall. And if you look at the x-axis down here, you will see circled in red PMA for Penium margaritaceum, and to the left of that, you have other green algae, and then we can move our way up into land plants, MPA being Marcantia, PPA being Physcomitrium, and you make your all the way up here to good old Arabidopsis. And what you'll notice is there are certain types, certain classes of cell wall processing enzymes that are found in very, very large amounts. I mean, Penium has a very distinct pectin cell wall, but it also has a very extensive, as far as we know right now, a group of enzymes that help model and remodel the cell wall. And a little better example of this is when we look at things like glycosyl transferases, we look at glycosyl hydrolases and pectate lyases, and you can see that Penium is right up there or exceeds what you find in some of the land plants. Another thing that we did when we were examining the cell wall of Penium was we treated the cells with stress. And one example was to look at what happens if cells are exposed to desiccation stress or osmotic stress, in this case, and also for very, very high light treatments. And the number of gene families that were upregulated by desiccation, just as an example, really went up. I mean, it was really quite impressive. And then there were a series of other types of genes that were also expressed. Penium is very, very sensitive about its cell wall. That's what we have found doing some experimental work. And that's why we think it is a wonderful model for looking at cell wall biosynthesis and the development of the cell wall and the cell. So what does the Penium cell wall look like? Well, it's pretty easy to get off. So you grow up this alga, it grows relatively fast. And then either by free shattering in liquid nitrogen or by sonication, you can get the cell walls to come off. And this is an example of a single cell wall. And on the outside of the cell wall, we have pectin. And on the inside, where it's a little smoother, that's where you have the cellulose. And this is this is a colorized SEM view of what that pectin lattice looks like. Okay, it's a series of fibers, some of them that extend outward, and it forms these projections. And we call it the lattice, even though it's not lattice in a true geological sense where there's very regular repeating subunits, there are repeating subunits. TEM examination shows the pectin here in the outer wall layer. We also see that this pectin layer is also attached to something that's a little less electron dense that seems to be embedded in this inner layer, which is made up of cellulose. So we did some immunogold labeling, and we used two antibodies, GIM5 and INRA-RU1, to see where we had different types of pectin. So in this outer layer, in this lattice layer, you have low esterified homogalacturona, that's calcium complexed. And then using INRA-RU1, which goes after the backbone of RG1, we see that's in these little roots that attach the outer layer to the inner layer. So we have been looking at other types of antibody and other type of probe labeling. And one that we would also be interested in is you could see these fine fibrils here on the outside, and we'll get to that in just a second. Those happen to be what we think is a arabinogalactan protein. And this is our very simple model of how the cell wall is put together. And here we're just looking at single polymers where we have cellulose and we have RG1 anchoring homogalacturonan. And we have the AGP molecule extending through the cell wall and attaching to the plasma membrane. But trying to put a composite of this together, this is probably a little easier to understand, where you have the homogalacturonan extending out from the cellulose cell wall, cross-linked by calcium, and it forms that very, very distinctive lattice. Now, this is nice, but we're more interested and became more interested in cell wall development. So one of the nice things about Penium is the fact that you can live label the cell with antibodies or other probes. And you can see, for example, here on the outside, when labeled with GIM5, this anti-homogalacturonan antibody, you can see this regular lattice of pectin, the extensions, these pectin that come up, fuse together and form the projections. Now, that's nice, but now you can use other types of probes. And I know Dan's lab uses this as well, reciprocal oligosaccharide probes. And one that has been very, very valuable to us has been the COS, the chitosan oligosaccharide probes. And this is where you are adding negatively charged you're adding positively charged chitosan fragments that have a fluorescent probe on them. They will basically hybridize, if you'd like, with the negative charge of galacturonic acid. And then you've got yourself a very, very nice probe, as you will soon see. So here's homogalacturonan in the way it is usually produced and packaged in Golgi vesicles. What you do is you take the C6 carbon, which is negatively charged, it's the acid group, and you put a methyl ester on it. So you basically neutralize the homogalacturonan. And once again, you do this because you don't want negatively charged things in vesicles. They never come out of the vesicles. So when the homogalacturonan is released to the outside, to the expanding area of the cell, you have deesterification occurring with an enzyme known as pectin methylesterase. And what that does is it gets rid of the methyl groups, and you have negatively charged galacturonic acid. So then what you could do is you could take a chitosan extract, a chitosan fragment, which is made up of glucosamine that has positively charged to it, and you line them up. And once again, these oligo saccharides that you have here are between 7 and 13 sugars. You also put a probe, a fluorescent probe on them, or you could put a gold probe for electron microscopy, and you can get some really, really nice results, even though the antibodies have worked really well. For example, this is what the pectin lattice looks like when you use these probes, and you could get to see so much more detail. And the obvious reason for this is that these chitosan probes are very, very small, and they can penetrate the cell wall much easier than these large proteins called antibodies. So being able to do live cell labeling with a variety of probes has been extremely important for us when we're looking at cell division rates, we're looking at expansion. So here's Pentium that's been labeled with GIM5, and Pentium produces new cell wall in an expansion zone that's found in its cell center. It will release pectins, for example, because we're going to be focusing on pectins here to the center. And what it does is it pushes the old pectin to the outside, and then the cell divides. So it'll be something like this. So in this cartoon down here, we have what's kind of a bipolar expansion. So this dark zone over here is where new wall material is being added. And the new wall material pushes the old wall material out as the cell expands. The cell gets to a certain size, and then it divides, and you have yourself two daughter cells. And theoretically, each daughter cell should be half new wall material and half old wall material. Sometimes that happens, and sometimes it does not. And that has been a very confusing point for us, which we're trying to investigate now. It can be a little bit frustrating. All right, so let's just take a look at how valuable this can be, especially when you're doing dynamic imaging of the cell wall. These are live cells. So on the left, we have the bright field imaging of cells that are being treated with cytochalasin. And then we have the correlative fluorescent. This was labeled with the COS probes. So I would suggest that you look at these two cells over here, though you're more than welcome to look at the other ones. And what'll happen is the dark zones represent where new wall material is being added. So what this allows us to do is to look at growth measurements, different expansion rates. We can also add a variety of different types of inhibitors to see how the cell wall disruption occurs. So using these probes has really been a fantastic thing for understanding pectin dynamics and cell wall expansion in this unicellular organism. As one of my students told me, what makes this great is because Pentium has in-your-face pectin. Okay, you don't have to go searching for it. And it's just a very, very nice tool for looking at cell wall expansion. However, there's something else in the cell wall, and that's adhesion. If you take Pentium cells and you wash off all of the gunk, and then you place them on just about any substrate, they stick to it like super glue. They really, really adhere. And at first we thought it might be the pectin on the cell wall, and that's not the case. So we wanted to look to see what happens. So here is Pentium that has just been placed onto a plastic substrate. And then within about an hour's time, they clump together. Okay. And if we use a variety of beads, okay, to look at what might be on the surface, we see that the beads stick to the cells when they've been washed and they've been put onto the surface. Okay, so penium sticks to things. And it kind of makes sense because for a lot of these green algae that are found in shallow wetlands, it's much more of a place to survive, a better place to survive if you're sessile, if you're attached to something, than being at the whims of floating in water. There's a variety of reasons, including forming biofilms with other microbes. All right, so what in fact is this stuff? What is this adhesive material? Well, we used an antibody, which is kind of the gold standard of monoclonal antibodies for identifying arabinogalactan proteins, and that's something called GIM13. If you label live penium cells with GIM13, you see that you get a surface covering, and it's kind of granular, and some of the granules will fall off, right? If we do co-labeling with a pectin antibody, GIM5, along with GIM13, we see that the GIM13 labeled area is to the outside. It's sticking on to the outside of the cell wall that has that pectin lattice. Doing immunogold labeling, we see that these fine fibers that are found, what we see with some of the immunolabeling, okay, also label when we're using gold particles. If we look at some high resolution imaging of what these fibrils are, they're roughly about 15 to 20 nanometers in diameter. They have a hollow core, and they're found next to one of these lattice structures, but not necessarily attached. This is TEM, and it's probably just a coincidence that they attach to it. And if we treat the cell walls with pectate lyase and remove most of the pectin, these fibrils stick. They stay. They're attached to the cell wall. And there's a reason for that that we will soon see. All right. So what are these things? You know, what's happening? So we take a cell, we wash it off, we place an individual cell on a substrate, we'll say a cover slip or a slide. And then we take a micropipette, we actually pull a micropipette and we roll the cell off. And then we label it with GIM13. And what you get left is a footprint. All right. So this stuff that's come off this particular cell, it's not exactly one-to-one, but it left it there. This is the adhesive material that's found on the outside of the cell wall. So in addition to having this unique pectin wall, there is adhesive material that's found on the outside. So what is it? All right. So we came up with another interesting feature, and that is, in addition to the stuff that's found on the outside of the cell wall, there's something else that's produced once the cell sticks. Okay, this also will contribute to adhesion, but it does something else. So you see all these cells at low mag, and what we've done is we've taken cells and we place them in growth media that has fluorescent beads. So you'll notice, you'll see a lot of the red cells here. That's autofluorescence from chlorophyll. Now, this isn't going to be something that you want to look at on a hot Saturday night, but look what happens. And there's two things you're going to notice. You're going to see that these cells are secreting something. That green stuff that's coming out, this fluorescent material, this is material that is being secreted by the cells, and they're attaching to the fluorescent beads. And then if you look in the background, if you look at some of the cells in the background, you see they're actually moving. This material is the extracellular pyrometric substance that these organisms will produce. Okay, and we'll stop this here. And we have a cell wall and then this extracellular material that really creates what we call the sticky business of penium and some of these streptophytes. Now, here's another video as to why is penium producing this extracellular material. Well, we have light that's been shining over here on the left-hand side, and I think it's going to be pretty obvious if we watch what penium is doing. You could actually see on the right, you can see some of the trails. So penium is secreting this mucilage, this gel-like material, this extracellular polymer matrix as it moves towards the light. So we have a photokinetic response with Pentium. So in this extracellular matrix, in addition to the cell wall, we have the sticky stuff on the outside, and we also have this EPS. So EPS is a common component of not only Pentium, but many algal extracellular matrices. It's found in things like Cavern and Nitella. It's found in Spirogyra. And what are the functions of these? People have looked into some of these mucilages for decades. We know now that it's used for gliding to get to optimal position for light. It's there for water retention, and it also forms a matrix for biofilms. Oftentimes, these microscopic algae are the keystone species in aquatic biofilms. And we've done some work with people at UCLA. And let me just tell you, the microorganism that we find in Pentium biofilms is extraordinary. And I'd be happy to talk to you about that, but we don't have enough time to do that here. So we isolated the EPS and the cell walls. And the important thing from this particular image right here is when we do a Western blot and we use GIM 13, we see that we have a couple of bands here that represent these arabinogalactan-like proteins that are found in the wall. And then we see a smear here. We're not certain as to whether the AGP that's found in the wall is being sloughed off or if we have AGP actually in this EPS. Okay, so, and then just very quickly, okay, I'll just show you a couple other things. So we decided that we're going to take a look at this adhesive material and the EPS, and we worked with Tony Bassick and Monica Doblin from La Trobe University, and we wanted to see what's in there. So in yellow, I've highlighted the key results. So if we look at the amino acid composition of the adhesive layer in the EPS, what we find are relatively large amounts of serine and hydroxyproline. And those are two very important amino acids that are in the job description of arabinogalactan proteins. Then if we look at the carbohydrate analysis, we see a lot of arabinose and other sugars, but the thing that has really surprised a lot of us is the amount of fucose, and we're not exactly sure what we have here with fucose. In fact, it was thought by a couple of other Australian collaborators that we might be dealing with fucoidin, which is more of a marine algal thing and not a freshwater. And then what are these polysaccharides that we found? We did linkage analysis, and we find that there's arabinogalactans in them. And also, once again, this confusing fucoidin-like material. And the last thing I could tell you about with respect to this adhesive material is that if you treat the cells with phospholipase and treat them for just an hour or so, you can remove all of this arabinogalactan material, arabinogalactan protein material, which is one of the factors which makes us believe that we're dealing with arabinogalactan that has a GPI anchor, which is typical of a lot of arabinogalactan proteins. This information on the sticky stuff, the sticky business of Pentium, will be out in another week or so in Physiologia Plantarum. So with Pentium, its extracellular matrix is really, really quite complex. And the individual components have some really unique functions, and it's been fun to play around with this. We did some genetic screening as well with Joss Rose and Eben Sorensen at Cornell, and wanted to see there were any homologs of AGPs, Pentium in comparison to Arabidopsis. And fasciculin, yes, we found others. Once again, not that you need to spend a lot of time looking at this, but the fact that we did find these homologs. We also looked at, for example, homologs for genes for that GPI anchor and also for glycosylation. And yes, we do have genes for the synthesis of the GPI anchor. We have proline hydroxylase genes that are there. So we do believe that we have arabinogalactans playing a big part in Penium. And what I could summarize is, oops. So what I like to hopefully, all right. So what I would like to summarize is that we are dealing with arabinogalactan proteins that are involved in primarily adhesion. They probably have a GPI anchor. They're not part of the pectin lattice. And we're still trying to figure out about fucose-rich polymers and fucoidin. All right. So the second thing that I just wanted to go through is one project that we're working on right now, and that is looking at shape formation, looking at how the cell wall responds to stress. So we know that the cell wall is responsible for promoting different cell shapes, whether it be root hairs, pavement cells of leaves, you name it. And the cell wall is critical in this. And at the macroscopic manifestation, you see this as phenotypic plasticity. So if in fact you, on a plant, if you put stress, you could have changes in the cell wall that will change cell shape, and it could change the macroscopic view of the plant as we have here in some Arabidopsis. And this oftentimes is associated with cell wall integrity maintenance. That is, the plant cell is constantly monitoring its cell wall. Obviously, the cell wall is extremely important for a plant cell. And if something happens to the plant cell, there are receptors on the plasma membrane that receive the message. And in the case of cell wall integrity maintenance, there are things, genes like Hercules, Feronia, Theseus, that might be these signal receptors. And they can send a message into the cell so that the cell might produce something like osmotic acid that's going to upgrade, it's going to increase, or it's going to cause the production of defense compounds. You may also have these signal receptors that are going to cause alterations directly to the cell wall, maybe through its biosynthetic pathways or through some enzymatic activity. And we were interested in the pectins of the cell wall. And if you're looking at an organism that has, once again, pectins in your face, it's Penium. All right. So we were interested in looking at Penium and the effects of its cell wall on cell shape. So as I showed you before, here's Penium. It is an elongate cylinder that has rounded edges, but this is Penium as well. If you treat the cells with cytochalasin, you get these cells to form filaments. If you treat them with the receptor-like kinase inhibitor roscotovine, you get this elongation in the middle. If you apply osmotic stress or salt stress to Penium, you get these very, very elongate cells that are viable, last for weeks. And all of this is recoverable if you wash out or get rid of the stress. Likewise, if you treat Penium with pectate lyase, the cells get very, very small, okay, about a third the size of what the normal cells are. If you use a microtubule inhibitor, which we believe is altering the microtubules in the expansion zone and affecting the composition of cellulose and trigger pressure, then causes it to swell, you can get this. All right, so Penium can take on a variety of shapes when you apply specific types of stress. But one of the more unusual things that we saw is if you treat the cells with pectate lyase and this microtubule inhibitor APM, what you end up getting is these dumbbell-shaped cells, which is really quite unique. And we were interested in looking at what is going on here. Are we looking at possibly Penium's response to being attacked with fungal enzymes? All right, so there were phenotypic changes arising as responses to multiple stressors. So just to give you just a brief background on the developmental mechanism involved in expansion and cell division in Penium cells, if you do microtubule labeling, you find a band that's found in the expansion zone. And what's unique about Penium is that shortly after the cell divides, it also starts to produce these satellite bands that are found on either side of this original band. And then these things move away from the original band due to expansion, or if you know anything about desmas, they may actually be actively moving on their own. So as Pentium expands, these satellite bands move to the daughter cells or the semi-cells, okay, on either side. When Pentium enters into cell division, the microtubular band that is found here in the center, it starts to reorganize and probably goes to the formation of the spindle. But these two satellite bands remain. And then when the cell divides, you have one of these bands that's found in the center of each of the daughter cells. This is very reminiscent of, first of all, the pre-prophase bands that are predicting the plane of cell division. But at the same time, what Pentium is doing, it's using these satellite bands to predict the plane of cell division in the daughter cells, which is quite unique. But it's these microtubular bands that are key. And if you disrupt these once again with APM, that's where you get changes in the cell wall. All right. So the expansion zone is where new cell wall material occurs. So as I mentioned before, here's the expansion zone. And then when you do your labeling, cell wall labeling, what you will see is that you'll have new pectin that's produced here. And where you don't see the cell wall label, that's where new cell wall has been produced. The cells will then expand to a certain size and they divide and you get two daughter cells. Okay. So now, what does this expansion center look like? Well, Pentium has a highway of cytoplasmic streaming, actin-based cytoplasmic streaming, just underneath the plasma membrane in the cortical region of the cytoplasm. And the Golgi bodies that are found in Pentium are found stationary, and they are found in lobes of the chloroplast. They do not move like many land plant Golgi bodies move. They don't move around and dock into ER. They're there, they're attached or associated with ER that is stationary. They produce vesicles that move to the cortical region. They get caught on these actin networks. They move around the cell. And depending on the developmental state, they get caught up in these microtubular bands. And they'll produce pectin and hemicelluloses. And then cellulose, of course, is woven in the wall. And that's something that we need to look at. All right. So if we treat Pentium with pectate lyase, these are live cells, you remove the pectin lattice. And what happens is that the cells get very, very small and you have remnants of the pectin. And then with Pentium, what happens is that the daughter cells have some unusual conformations, but they're subtle. They're not very, very prominent. And that just kind of indicates that cellulose is the load-bearing component of the cell wall. But more importantly, and the last experimental thing I'd like to show you is, okay, well, first of all, we know that APM, as I showed you in a previous slide, causes this swelling in the middle. But then when we apply pectate lyase at the same time, we have the swelling in these opposite sides, giving this dumbbell-shaped cell. What we think is happening is that Pentium has now cut off expansion and cell division in this zone in the center. And the satellite bands of microtubules are marking the zones of new expansion centers that are affected by the APM. All right, but then there's something else. And once again, I showed you, it's these bands over here that are probably the zones where this happens. But something else. What's happening in the central zone over here? We think what Pentium is doing, it is basically doing a burnt ground policy type thing. What it's doing, it's cutting off this original zone. What happens is you have a retraction of the cytoplasm. You see these little ellipticoid bodies, and these elliptical bodies are Golgi bodies. And one of the labels that we use for identifying Golgi bodies is a dye called MDY64. It's really good for looking at Golgi bodies. And what you find is that there's a clustering of Golgi bodies in this zone where you used to have expansion, but it looks like it's going to be a cutoff zone. So here's a TEM micrograph showing you what that looks like. All right, and here's this zone right over here, and the nuclei are starting to migrate away. But if you look at this zone just to the right of the central zone over here, you see all that cytoplasm. But more importantly, if you look in the center, you've got this growth that cuts off the cell, that is a different type of cell wall material that is cutting off the cell. It's almost like what you have in pathogenic responses in some plant cells, where you use these cell wall plugs to stop a particular pathogen from coming in. So these are formed by a furrow. Okay, these this sort of papillate, or whatever you'd like to call it, the sort of incursion, and you have a furrow probably formed by the Golgi vesicles that will form this thing that's going to cut off the cells. You do calcifer labeling, and we assume that this is cellulose. So we thought, okay, well, oftentimes you'll find callose as being a thing that is going to cut off various regions in a pathogenic response. It's not. We have these arabinogalactan-like components that are found along with some pectins, but we use the antibody CCRC M80, which goes after these arabinogalactan and arabinogalactan-like proteins. So it's a very, very different type of incursion that Penium is producing. And just to show you that if you place these cells back into regular growth media, you wash them out, what'll happen is this used to be the cutoff zone, and then new cell wall material starts to form at the opposite end, which is also an interesting situation. All right, so in this last thing I wanted to talk to you about, we see that cell wall plasticity and cell expansion and morphogenesis may be induced by changes, okay, to the cell wall. Changes in shape and changes to the cell wall go hand in hand. And this phenotypic plasticity may be an advantage to an organism coping with various stress agents. If you're living at the interface of a terrestrial and an aquatic ecosystem, you are facing water, you're facing salt stress, and being able to form different shapes, maybe even becoming more filamentous, going back to your evolutionary past, that might be an advantage. So what are some of our future investigations, okay, with Penium and other streptophyte algae? Well, we want to look a little bit more at the extracellular matrix, especially the arabinogalactans and the fucoidins. The fucoidins have really piqued the interest of Tony Bassick, and Tony is a leading cell wall biochemist, and he just doesn't believe what we have there, and that's what we need to look at. But another type of project that we like to look into is the Golgi apparatus. And I wish, there was time to talk about it, but I got to tell you, Penium is a lean, mean secretory machine. It has 150 to 200 Golgi bodies per cell. As I mentioned, they're not moving around in the cell. They're stationary in these valleys of cytoplasm. They produce these vesicles that move to the peripheral cytoplasm, and then, of course, they're sent to expansion zones or wherever. Well, one of the unusual things that we've noticed is that when Penium is putting out these hypersecretion events, when it's producing that extracellular matrix, it looks like the Golgi bodies, and we're pretty certain it looks like adjacent Golgi bodies fused together. It's more like what you see with the ribbons of Golgi bodies that you find in many animal cells, where you do have fusion when they're producing large amounts of a product like collagen. So one of our goals, and this is the last thing I want to show you, is we're trying to create a Penium lattice, and Josie Larico, who's my postdoc, she's the one responsible for this, and she's trying to come up with this beautiful atlas and putting everything together. So you'll see all the different organelles that are being added. This happens to be one of those treated cells, but our hope is to come up with complete three-dimensional profiles, structural profiles. And the tool that she uses, array tomography. And there we go. Ta-da. So thank you very much for listening to my talk. And I'd like to thank Josie, Lily, who is our microscopy guru, Katya, who was a postdoc at Skidmore, a whole range of undergraduates that you see listed here, our research collaborators from many parts of the globe, Cornell, La Trobe, Ireland, funding from the National Science Foundation, And this picture that you see over here is AI generated. My colleague at Cornell, Jocelyn Rose, who's Mr. Tomato, he thought it would be great to show this tomato coming out of this ooze of water to show that where land plants came from. So thank you very much.
Thank you. Thanks very much, David, for a wonderful talk. It's super nice. So if you have a question, why don't you use the little hand raise tool, and we could scroll through and just shout out with your microphone that you got a question. Then if you turn your camera on, David could see who he's talking to for the question. So are there any questions?
Yes.
Here we go, Josh.
I think Sharon was first.
Okay, Sharon.
Hi, that was a great talk, a really interesting system. Can you do genetics with these? Can you knock genes out and look for
Yeah,
function?
that's been a challenge. For example, we've never had success in transforming these organisms. Currently, there's CRISPR-based experiments doing knockouts, RNAi, and the problem with these organisms is that they're very resistant to a lot of molecular manipulation. A couple of years ago, we had a symposium for streptophyte algal biologists in Spain, and we had this one session on what's the most difficult thing with working with these organisms. And we've just had very little success so far in doing any type of genetic manipulation. Recently, though, in Japan, there's a group that believes they found a way to transform, in fact, Penium, and they're using something called pulse electroporation. Um, whether that works or not, and whether other groups have, I do not know, but it's been a problem.
I have one more question. So those microtubule bands that you see forming reminds me of the pre-prophase band. Does it
Yes.
share characteristics with higher or angiosperm or other plants?
Oh, yeah, we think they're very much like pre-prophase bands, except what happens is the microtubules will reorganize when the spindle is forming, and that's why I was saying that that's probably where the microtubules go. You just don't disappear like a pre-prophase band does. But the thing we think is cool about Penium is the fact that you've got these future pre-prophase bands that are produced and are sent over to the daughter cells.
Oh, cool. Thanks.
Josh, go ahead.
Yeah, great talk. Very beautiful slides. So I've asked variations of this question before, and I'd like to hear people's thoughts on it about, why this happened in the green lineage, and we haven't seen the red lineage make this similar type of transition. And maybe it's just because the green has already conquered that niche. But do we see anything in the red lineage that might look like it's in a transition or collecting some of the apomorphies that you showed, phenylpropanoids, things of that nature, that would be needed to make the transition onto land?
I really can't answer that. I really don't know. The red algae have been a group of algae outside of the applied aspects with agar and carrageenan and all that kind of information. They've kind of it's the amount of research that's been done is far less than what you find with green plants.
Mhm.
And I think what a lot of researchers have found with the green algae, when you find organisms just like I've talked about, but then also you find things like Chlamydomonas and Volvox and Chlorella and all these great model systems. It's been the one that's elicited the most interest, but I do not know much about the red algal lineage.
Thank you.
I've got one for you, David. It pertains to the slide where you showed the AGP structures in a pectinase treated cell. I don't
Yeah.
know if you could go to that slide, but I was just curious about your thoughts about how that structure could assemble, because the length of it was on the order of almost a micron or half a micron. it's
Mm-hmm.
seen. And then the proteins on the gel are, I don't know, 100 kilodaltons or so. What are the thoughts about, yeah, that lower right? Like, how do these
Yeah.
fibrils form? And how do they generate these lengths that approach the micron scale?
All right. Well, we know that they're one of the first polymers that is placed onto penium during expansion. So when penium is producing its new cell wall in the cell center, the first thing that you'll see two polymers, maybe more, but two polymers, the cellulose, and then these AGP-like fibers that are being formed. In the cell center, they're smaller. They're only about half this size. And then as you start to move towards the polar regions, they start to expand. So as the wall grows, these also start to increase in length. And those are just observations. And but we know they do not have anything to do, well, have very little, if anything, to do with the pectin lattice. They might have some physical connections with it, but yeah, it's a good question, Dan. I'm just giving you what we observe and can't really go too much further than that.
Okay, all right, thank you. Cindy, you've got one.
Yeah, there's a question in the chat from Dewan Wang. She is with Dr. Zhu's lab, and she has two questions. As the original of land plants, does pinium have any function related to pathogen response / fungal interaction, etc.? The second part is, has any genes identified been applied to advanced plants? Do they have the similar function in the advanced plants? Dewan, feel free to go ahead and unmute, and you can add to that question if you have anything.
Yeah,
Yeah.
I can explain a little bit. So the first question is, I'm interested whether the fungal related genes is involved before the algae cell or before the plants colonized from the water to the lands, or it's involved during the plants after they colonized onto the land and then involved later on. And the second question is, I'm just curious about how similarity between the algae genome to advanced plant, and is that possible if we applied the algae genes into the advanced plants for the function verification?
Okay, so to answer your first question, in the laboratory of Jan de Vries, he's in Europe. His last name is spelled Devries. He has looked into possible associations of streptophyte algae with fungi and possible symbiotic fungi and possibly mycorrhizal fungi. And while there's not a lot of evidence, he's looked into the genome, not penium so much, but into some of the filamentous zygnematophytes. And he believes that that there is evidence that these organisms probably form symbiotic associations or associations with fungi, which most land plants do, and maybe even mycorrhizal-like. And in fact, I'll show you. Wait a minute, hold on 1 second here. And one of the problems is getting old and having to wear glasses. Okay, what I'll have to do is I could send you the reference. It's in the Annals of Botany. It's in 2024, and the authors are Jan de Vries and his wife and a few others. And what they do is they go into the phylogeny of the streptophyte algae. And one small section there is on possible associations with fungi and bacteria. And that will certainly give you at least a starting point to look at the evidence that suggests that there may have been associations before plants got onto land. Okay, and then with respect to your second question, there appears to be quite a few similarities in the genome that you find with some of these algae. and also with land plants. I mean, in one area that I would suggest looking into, where there's been a lot of work, has been that in hormones. Okay, and genes for the pathways leading to things like jasmonic acid, abscisic acid, and especially ethylene. And interestingly enough, you could probably find the answer to that in the same paper. It was a really very nice review paper that just came out in Annals of Botany. So this answer to your second question is, yeah, there's some similarities. And that's probably the best source that I could tell you to look into.
Okay, thank you.
Sure.
Any other questions? Going once, going twice, thrice. Well, let's send a big clap out and thank you to David. So thank you so much for the nice seminar. Really pleased to have you come. Thanks. So David, if you got a moment, you can stay on just for a second after we're done.
Yeah, no problem.
All right, thanks everybody.
Okay.
Awesome, thank you.
Oh, David, that was great. Thank you. I wanted to ask you one more thing about this COS 488 labeling. So
Okay.
So when you're doing those experiments,
You're welcome.
did you wash the probe away? And that band, I was surprised that in that band it stays black. And is that because it was like a pulse labeling with the probe?
No, no, in that zone that was black, that probably was where you had a esterified pectin being secreted. There's a narrow zone in the expansion zone that you where the COS probe doesn't bind to, because the homogalacturonan that's secreted there still has probably a sufficient number of methyl ester groups blocking the binding of the chitosan oligosaccharides. And then also, if you move out from that central band, there are some zones where you also see no labeling, and we think they're growth oscillations. We think that pectin synthesis takes place during the day, takes place during the light, and then at night it slows down or shuts off, and then you see these little bands. So I just think what we're seeing in these dark zones is where you still have esterified homogalacturonan.
Mm-hmm. And in the extended time lapse where that band became very wide, I was just surprised that no new probe appeared at extended time points when that band was quite wide.
Yeah.
So
was I.
The probe is still there. It can't bind.
Yeah.
Huh.
Yes.
That's so weird, because the geometry of that band changes over time. It's getting larger. So it's weird that it doesn't rebind.
I see what you're saying. Yes.
Okay, cool. Well, thanks again, David. And I'll be in touch.